I'd like to introduce Russ Weeks. He's already given the industry keynote today on Accumulo in the enterprise. He is a software architect at FEMI. Yeah, I wanted to call it FEMI. It's FEMI. And he, prior to joining FEMI, he worked in the network management groups at Ericsson and Cray supercomputers, where he discovered a passion for distributed data structures and algorithms. I hope you enjoy his talk. Russ Weeks.
Thanks. Hey, everybody. Thanks for coming. I wanted to use this opportunity to share some of the things that I learned while I was working on one of the most fulfilling projects that I've had a chance to be a part of in a long time, which was designing and building a large distributed warehouse for storing genomic information on top of Apache Accumulo. So I remember being here last year and being in front of you guys, and I gave a talk last year about agriculture, and I opened that talk by saying, I'm not an agronomist, and I'm going to open this talk by saying I'm not a bioinformatician, but one day we'll figure out what I am, and that's going to be a fun journey. So I'm going to start by doing my best to describe what a genomics warehouse is, a data warehouse is, what kind of questions we're trying to answer, and then I'm going to go into more technical detail about why it makes sense to build a genomics warehouse on top of Apache Accumulo. But to start with, I want to run through some definitions. So just like a really fast introduction to bioinformatics from somebody who, again, is not a bioinformatician, but I think I'll define these terms sufficiently for the context of this talk, but don't go out and try to sequence your own genome based on what I say here. So everybody has a genome, right? So let's start there. Your genome is the total of your genetic material. Your genome consists of a sequence of bases. If you've ever seen the movie Gattaca, if you remember that movie, the GTAC, packaged in a set of chromosomes. Some chromosomes are represented by numbers, 1 to 22, and some are represented by letters X and Y. So physicians and researchers, and those are the two groups that we're going to focus on today, are usually most interested in the way that a person's genome differs from a reference human genome. And how you define a reference human genome is an interesting question in its own right, but today let's proceed with the understanding that there is a baseline, and doctors and researchers are generally most interested in how you differ from that baseline. So these diffs are called variant observations or variants. And we can describe a simple variant in plain English by saying something like, on chromosome 15 at offset 1,250,326, there's supposed to be a C, but you have a T. And so your genome probably has between 4 to 5 million of these variants. And the vast majority of them are meaningless. Many of them are unique to you and have never been observed before. These variants are the noise in your genomic signal. Other variants are important. Or clinically significant. Some clinically significant variants confer benefits. For example, if you're missing 32 bases around position 46 million on chromosome 3, you might be immune to HIV. Unfortunately, many significant variants are pathogenic. They might lead directly to a disease like muscular dystrophy, or they might put the carrier at increased risk of disease like obesity or Alzheimer's. This is the depressing part of this talk, because the more you get into this, the more depressing it gets. And I never really understood the phrase ignorance is bliss until I got into this. Like, I don't ever want to get my genome sequenced, if I'm being honest. So we have these variants. We can tell the ways in which you differ from a reference human genome. The question is, what meaning does each of those variants have? And this metadata about variants, has it been identified before? How significant is it? How frequently do we see it? And so on. This metadata is maintained in reference data sets. So let's take a quick look at an end-to-end genomics pipeline, and then we'll focus on the later steps in the workflow where the storage and analysis of the variant information starts to come into play. It starts off with a tissue sample, right? It turns out that different types of tissue can have different genomes. Cancer cells specifically have a pretty crazy genome, and it's very instructive to see in what ways does a genome from cancerous tissue differ from a genome in non-cancerous tissue in the same person. So already we have a one-to-many relationship between one subject, that is to say, one patient, and many samples. So genetic material from the tissue sample goes into a sequencing machine, and it pulls out a bunch of little strings of those bases that we talked about before, combinations of G's and T's and A's and C's. This is your genetic material, but there's no context to it yet. We don't know where these little snippets fit in your genome, and that comes in the next stage, which is called alignment. So in the alignment stage, an algorithm tries to sequence or connect these overlapping strings of bases. And one metaphor that's really stuck with me is that if you imagine somebody takes a few copies of the works of William Shakespeare and cuts them up into little tiny fragments of text, and at the end and gives them to you, and having no knowledge of how they're connected or the works of William Shakespeare, you need to overlap the snippets of text in such a way that you get a result that is the works of William Shakespeare. So again, that is a fascinating problem. It's a super interesting computer science problem. We're not going to talk about it. We're going to assume that some other thing has solved that problem. So after the alignment stage is finished, we figured out the genome for the input genetic material, and then what's next is figuring out those diffs relative to a reference genome. This process is called variant calling, and the output of this process is variant call file, or VCF. And we're going to come back to the VCF in a minute because that's where it starts to get really interesting from a storage and indexing perspective. But before we get there, I just want to address the last step in the process, which is annotation. Annotation is where we take some known facts about certain variants and associate them to our observations. So we know that you have a certain variant, but what does that mean? How many other people in a population have the same variant? How many people in a population that shares your ancestry have the same variant? Is it benign or pathogenic? So often this metadata comes from publicly available data sets. The National Institute of Health primarily has a huge, vast volume of genomic resource information, but a market is starting to emerge for private reference data sets as well. So on one side of that variant observation, we're linking that genetic address or locus of chromosome, offset, reference, alternate to known metadata. And on the other side, it's very common to link the sample ID or where this observation came from to clinical data about the subject, ancestry, clinical history, demographic information, treatment regimes, and that kind of thing. So the last thing that I want to explore before we start getting into the technical detail is who uses this data and what do they do with it. And I want to focus on two types of users who have similar but slightly divergent goals. One group consists of researchers. These people want to build patient cohorts. They want to combine clinical data and genomic data to find people who share certain traits. And so a simple example might be, find me all men between 35 and 49 who have a high mutation burden in the FOXRED1 gene. On the other hand, we have physicians who want to understand a patient's genetic makeup in order to make treatment decisions about that person. And this is really the foundation of this emerging discipline of precision medicine. And what these two groups of users share is the need to move back and forth between observed variants, known variant metadata, and clinical or phenotypic data. And this is really, this ability to navigate between these three spaces is sort of the core feature of this data model that we're going to look at. So that's a high-level overview of a genomics workflow from a wet lab to a data warehouse. We're obviously at the Accumulo Summit, so I'm talking about a data warehouse architecture that looks like this. The data is going to start as flat files in HDFS. The variant data is annotated and indexed in Accumulo using a data model that I'm about to describe. And then it's accessible by researchers and doctors directly in Spark or through a nice notebook and visualization environment like Apache Zeppelin. So let's get back to that VCF file, which is really just a fancy TSV file. So again, the purpose of the VCF file is to track your diffs relative to a reference genome assembly. VCF is probably the most common format for sharing and indexing genomic data. So we'll take a quick look at an example, which is that's an eyesore. The first thing that jumps out is that there's some header information there, and I'm not going to really talk about it too much, except to say that the format is a little bit self-describing. So let's focus instead on a variant observation. So here we can see a particular sample identified as NA0001, and we can see that at offset 14,370 on chromosome 20, there's supposed to be a G, but this sample has an A. So this is that single variant observation that I was talking about. So what I'm trying to build towards here is that we've got the beginnings of a data model. So every variant observation can be uniquely identified by the tuple of values sample ID, chromosome, offset, reference, alternate. In database terms, we'd probably call this a natural composite key. And I think you can probably see where I'm going here. We have a key that uniquely identifies a tiny bit of genomic material, genetic material, and we have a database that's really good at storing billions and billions of small key value pairs. So if that's the structure of our key or the structure of our row ID, this is what I was getting at earlier when I was saying that we can link different parts of the key to different sources of information. The sample ID is, you can think of it as a foreign key that sort of unlocks a wealth of clinical information. This could come from an electronic medical record system or a healthcare data model. And it's typically very tabular, relational in structure. And the rest of the values that form that locus, chromosome, position, reference, alternate, can potentially tell us what researchers around the world have learned about this variant. So this is a great candidate on that right-hand side of the key of the tuple of values there. It's a great candidate for denormalization. I mean, you can imagine how the naive approach of joining two tables together falls down really quickly when you have one table with tens or hundreds of billions of rows and another table with maybe 200 million rows, and very frequently an inner join between them produces almost nothing because of that problem that most genetic observations out there are unique to you and have never been seen before. So it's pretty clear that this tuple of values is going to form part of our key, but let's consider an alternative before we go much farther. What if we put the sample ID at the end of the tuple rather than at the beginning of the tuple? Does anybody want to hazard a guess as to how that affects our data model and how we can query the data? What's that? Pardon me? Yes, it does. It affects the sort order of the tuple. And in doing that, in the first form of the key, all of the variants for a certain sample will be grouped together logically. They'll be adjacent to each other. And in the second form, all of the samples in which a certain variant was observed will be grouped together. And what's interesting is that those two models correspond exactly to the two groups of users that we were talking about, right? As a doctor treating a patient, I want to be able to look at a patient's genome or part of a patient's genome and make the decision, like, is this medication going to kill them or not? And as a researcher, so in that case, I know what sample I'm looking for, and I know what part of the genome I want to isolate. As a researcher, I'm much more interested in saying, okay, show me all patients that have that share a certain variant or a set of variants. So my understanding is that most genomic warehouses store data in the second form, where the variant is, language of data warehousing, the variant is the fact and observations of the variant are a dimension. So that nails the researcher's use case, but I think it makes it very difficult to address the doctor's use case. Putting the sample ID first at the beginning of the tuple gives us nice hierarchical layout to the key. Sample has many chromosomes, a chromosome has many positions, a position has a reference allele, a reference can have many alternates. I like it. It pleases my sense of design. It satisfies the doctor's use case, and in a sec I'll show you how it can satisfy the researcher's use case as well using some components of Accumulo. So now our key value model is starting to shape up like this. If we imagine this variant attribute table where we can have one variant observation and a bunch of properties associated with it, right? This is pretty standard entity attribute model that we can see in Accumulo. Obviously, there's a bunch of stuff that I have to gloss over here just in the interest of time. Like, for instance, how do you encode the values and what's the best way to serialize a composite key, but this is a good start. So you can see the tuple of values forms the row ID. The attributes of the variant form the column qualifier if we're being fussy. And then the value of the property goes into the value field. And so this key schema, like I said earlier, this key schema is going to be very good at addressing queries that specify some or all of that key hierarchy. And as it turns out, that's a very broad set of queries. It's going to let you answer questions like, does this patient have a variant that will cause a certain medicine to kill him? Or what differences do we see between this patient's cancerous tissue sample and his non-cancerous tissue sample? And it's very useful to answer those questions. But it's not going to get us very far with a question like, show me all samples with pathogenic variants in a certain gene, or show me all observations of a specific variant by its dbSNP identifier. dbSNP being a public reference data set. And fortunately, though, there's a very well understood, commonly used data structure in Accumulo that can help us answer these questions, and that's the transpose table. Every single talk that I've been in today has talked about some twist on the transpose table, so it's clearly a very widely used feature. So for our purposes, let's call it an attribute variant table. We're going to flip those key value pairs around and structure the table like this. Again, I need to breeze through this, so I'm not going to go into detail about what you could use the column family for, or the column qualifier, the value fields. Obviously, we have the flexibility of defining certain locality groups with the column family. But you can use placeholders for them, and you go a long ways. So those key value pairs get transposed into something like this, and I just want to make the point that they're not adjacent. In this table, they are adjacent to each other, and in this one, there could be millions, potentially millions or billions of key value pairs between them. But they get flipped around and look like this. And so those previous questions that we couldn't answer, we can answer very effectively with this model. Questions like, show me all variants for a particular known variant ID. Because the attribute values are ordered, and because they include pointers back to the variants. And in many cases, that pointer back to the variant is all you need, because it contains useful information. It's a business key or a natural key. We can tell from that this variant was contained in this sample, or so that it applies to this person. Or we can tell where in a particular sample it existed, so we can see if it's in a gene that we're interested in. So there's often no need to go back to the variant table to get the further metadata that we know. So the last thing that I want to say about the transpose table here, the attribute variant table, is that we get a bit of a pass. There's been a lot of talk about how do I keep tables like this up to date in the face of a constant stream of updates and inserts into my database. That's not such a big deal here, which is nice. Took a bit of a load off, because genetic material is pretty much immutable. It stays with you for as long as you're around. And it gets updated sort of in batches when new people get added, when new data sets get added. So we don't really need to worry about keeping the transpose table up to date with the data table in the face of rapid, constant, high velocity change. So the transpose table was one killer Accumulo feature that we're going to use a lot in this data model. Another feature that it makes sense to use very extensively is bulk ingest. So we're getting our input in batches of roughly 5 million rows, or roughly 90 million key value pairs at a time. And there's no need for us to fire mutations at a tablet server as fast as we can. We don't need that low latency ingest and then lookup behavior. So we've had a lot of success using Spark, using a Spark job to take that VCF file, that tab-separated data file, and generating the R files that go into different tables. So we store the data in both those schemas that I showed earlier, and then we also store it in a summarized form. And this is the step where we would annotate all of the observations in the VCF file with properties from known data sets. So the trickiest thing to get right is aligning the R files for the transpose table with the split points of your table. In Spark, if you use the default hash partitioner, and I think this is a problem that everybody hits, certainly everybody that uses Spark in Accumulo, I think, and probably many people that use MapReduce in Accumulo as well. If you use a hash partitioner to sort of organize the work between your executors, you wind up in this worst case scenario where every tablet server owns a piece of every R file. And you'll know that you're in this. Well, that's fantastic. You'll know that you're in this situation because your cluster is just compacting like crazy, nonstop, and your bulk ingests start to get stalled because every chunk of your key space is referenced by every R file in your system. So it's the worst case scenario. So you mitigate this problem by taking a look at the split points of your table, of the transpose table, and then using a Spark range partitioner instead of a hash partitioner to kind of match them up. So that's the outcome that you want. And it doesn't have to be perfect. It just sort of has to be better than the pathological worst case. And you can go a long ways. And so we, before we had this, before we figured this out, when we were still using the hash partitioner, ingesting that 5 million rows, 90 million observations was sort of getting up to minutes, tens of minutes for a small cluster after we'd sort of loaded up the cluster with a bunch of data. And afterwards, it's rare to see a bulk ingest that takes more than 5 or 6 seconds. So another Accumulo component that we use to work with this complex key structure is the seeking filter. Seeking filter is an iterator that's available out of the box in Accumulo 1.8. And probably the best way to describe what it's good at is to compare and contrast it with the interface of the filter iterator. So if you've written Accumulo code, that's probably familiar to you if you work with iterators. Filter iterator lets you define a predicate in a subclass, and then as the key value pair progresses up the iterator stack and it hits your filter, it will only move further up the iterator stack if the predicate returns true, right? Pretty simple. So the seeking filter allows subclasses to test key value pairs as well, but in addition to passing or blocking a particular key value pair, the seeking filter can also give a reference to how far the underlying stream of key value pairs should be advanced. So if you can tell, if you're in your seeking filter and you can tell by looking at a key value pair that maybe you're not interested in the rest of the versions of a particular column, or the rest of the columns in a particular column family, or the rest of the columns in a row, or even up to some arbitrary lower bound that you can determine yourself, then you can pass that information as part of the response from your predicate, and then the seeking filter parent class will just sort of advance to the next thing that you might be interested in. So it's a great fit for some of our query patterns, the query patterns that kind of look like this. So if you say, for example, a researcher might be interested in all variants in a set of genes. So if I have sort of 50 genes that I know are correlated with stomach cancer, and I have 10,000 samples that I want to inspect, you could imagine, based on the key schema that we talked about, how you could take the product of the genes that you're interested in, like chromosome 15, position 2 million to 2,100,000. That's one gene range potentially. So you can imagine how you could take the product of all of the genes that you're interested in and all of the samples that you're interested in, figure out the scan ranges, pass them into a batch scanner, have it do the work. But that act of taking the product means that you can get this explosion in the number of ranges that you're interested in. So alternatively, you can use, for instance, the seeking filter to get that stuff without having to figure out the ranges up front. So it's also good for sort of unbounded ranges where I don't, for instance, know all the samples that I care about. I might just say, I care about this in every sample. You figure out how many there are. And it's also better than a brute force approach where you're just scanning the whole table and throwing away the key value pairs that don't match your criteria. So it's really good for when you have a bunch of small ranges that might be too difficult to compute on the client side, or too onerous, but still constitute a very small quantity of your overall data. Right, so I'm going to step through a bit of code. This is not exactly a great format to share code, but if you'd like, there's a gist at that URL. That's an O at the end, a capital O, if you want to follow along. And the nature of the code is to solve a problem that I've encountered over and over and over again. And I think is based on how often that idea of a transpose table keeps coming up, is probably something that a lot of people are seeing. And that is, how do I go from rows in a transpose table to rows in my data table? Like what's the right way to go from those row IDs to where the row IDs came from? So we saw in the Presto talk, that work was being done in the Presto workers. Alternatively, that you could say, well, I'm not going to. I'm going to make them the same thing. I'm going to use, for instance, a partitioned index or a sharded index, so that my transpose values and my values are actually in the same tablet, and I can use a tablet server to go back and forth between the two spaces. But one approach that doesn't scale well is to take all of the key value pairs from your transpose index, bring them into one place, one centralized place, and then try to dereference them against the data table. So there's a really clean way to do this in Spark. If you're using Spark and Scala, it works out to about 50 lines of code. 50 lines of code because it's a Kerberized version. It would probably be about 25 lines if it wasn't. And so you can go through the gist there, but I'm just going to walk through the main points. And these are the main points here, is that so you scan a range on your transpose table. So that might be all variants of pathogenic clinical significance. And that's going to give you a bunch of references into your index table. And they're going to be scattered across your index table, right? They were nice and adjacent to each other in the transpose table, but they could be scattered from the top to the bottom of your data table. But you're going to use Spark to get that. You'll get an RDD of those rows. And again, if you're familiar with Spark, it won't actually be a data structure that contains those rows. It will be an item in the directed acyclic graph of computation that says this is how I'm going to get them. You call repartition and sort within partition on that RDD, and I'll get to why in just a second. And then you map the partitions of that RDD. And then in each of the partitions, the first thing you're going to do is set up a batch scanner. And then while there are still items in that partition to consume, you pop some configurable quantity of row IDs out of the partition, and then you feed that batch into a batch scanner using exact match ranges. Say I'm interested in these rows, this row and this row and this row and this row. And so you do that over and over again for maybe 100 or 1,000 or whatever a reasonable sized number of batches is to put into a batch scanner, because it's probably not 1 million or 2 million or however many hits you got on the transpose index. And then you yield the output from the scanner as the return from that partition. That second step there, repartition and sort within partition, it's not strictly required. The code will work if you don't have it. It will work 14 times slower than if you do have it, was our measurement. And I'm not sure why that is exactly, but I think it's because it takes advantage of Accumulo's caching more effectively. If you can order those rows that you're looking for, then you can take advantage of the look-ahead cache in Accumulo in the tablet servers, because it won't have to go to disk as often as it would if it were just sort of randomly grabbing rows. So the last thing that I want to talk about before we get to a quick demo of this model is the security model that we're going to work with. So we'll talk about access control for a second. There are obviously implications here for patient privacy, right? A patient must give their consent for their genomic data to be used for any purpose other than primary care. What I didn't get when I started this project was that people also want to control access to those reference data sets. A lot of this research is carried out in an academic context, and I kind of had the impression going in that academia is this place where information flows freely and people exchange knowledge all the time with each other, and it turns out that's not always the case. Researchers can be extremely protective of their data, especially of unpublished data. And furthermore, my initial assumption was incorrect as well. This work isn't always being done in an academic context, and there is a profit motive to license some of this data to licensed users. So we're talking about horizontal access control. These are patients that have released their genome for specific research purposes, and also vertical access control, because the annotation data sets, the reference data sets, can be licensed to certain groups or organizations, right? And that yields a security model that sort of combines the two. A really clean way of satisfying both those requirements is to and the two together. So you have an authorization expression that controls access to the row level based on patient consent, and to the column level based on data licensing. So let's get into a quick demo. I have a small system set up in AWS. It's a 4-node cluster, 64 samples from a public data set called the 1,000 Genomes Project. So this is just public information. Altogether, it works out to about 400 million variant observations, or rows, and 7 billion attributes, or key value pairs. But we've verified this architecture up to tens of billions of variant observations and nearly half a trillion key value pairs. So it's pretty solid. So let me just change really quickly to show that. Nope. It's not on that side. There we go. Yeah, that's better. Okay, so that's the that's the gist, and I'm glad that I didn't walk through in front of you guys, because that's a bit much to take in on a small screen like that. But it is, I mean, I lied. It's 77 lines of code to do that distributed dereferencing of row IDs from a transpose table to a raw data table. So it's very compact, it's very easy to use, and it solves a problem that I personally run into all the time. So hopefully it will be useful to other people as well. Okay, so this is Zeppelin. If you've never seen it before, it's an interactive notebook environment. It's a lot like IPython notebooks. It's an Apache project. I'm not sure how relevant it's going to be to this community, because it is one of those projects that I was referring to earlier that assumes, at least in its current version, it assumes that the entire cluster can be turned, or a large chunk of the cluster can be turned into a shared Spark context for use by different people. And data frames and RDDs and tables in a Spark context can be available to anybody that has access to the Zeppelin server. So you're sort of faced with this idea of, do I want to, in a secure environment, in a secure processing model, you're faced with this idea of, do I want to subdivide my cluster and give different people or different groups Zeppelin servers, in which case I'm sort of losing the distributed power of my cluster, or do I want to give everybody access to a great big Zeppelin server and let them share data? There's pros and cons to each model. And I think it's going to get better. In the next version of Zeppelin, they're introducing an adapter for a project called Livy, which has finer-grained access control inside the Spark context. So we'll just walk through a couple of these paragraphs to start with. A couple of common queries that I think, again, these are sort of typical of the types of queries that doctors and researchers are going to want to do. So this is, let's find all pathogenic variants in the collection. And again, there are only something like, how many did I say? I think there's a few hundred million, 340 million rows in this data set, something like that. So it's not big, but it's big enough, I think, to prove a point. So if we run this, we can see that it comes back very quickly. This is not using cache data. This is just hitting the Accumulo tables directly. And we're getting 367 results back in sort of sub-second response time. And so this is, I mean, I don't want to, I work for a proprietary commercial company. We offer this as a commercial offering, but I don't want to sort of say this is how wonderful it will be if you license our product. This is using the schema that I talked about earlier, the key schema that I talked about earlier, and the table structure. And this is a query, just clinical significance 5. That's pathogenic stuff. That's stuff that is likely to kill you if it appears in both of your chromosomes, both pairs of a chromosome. This is just hitting the transpose table. So we have the information from that transpose table, and it's not going back to the variant table itself. Right. Just the last thing I want to point out. This ClinSig contains 5. That's just a trivial little DSL that we put together because we got really sick and tired of trying to hand code ranges on the transpose table. But you can imagine it's taking that statement and it's turning it into a range, a single range of values that it can scan on the transpose table. So that was just looking at the transpose table, but let's see what happens if we want to actually dereference the data against our index table as well, or against our data table. So we can see, again, performance is very good. We're dealing with trivially a very small data set here, but what that's doing is it's going through that chunk of code, and it's going for each of those values in the transpose table. Let's go and see. Let's turn that into an exact match range on the data table and look that stuff up. And we can see that now we're getting a lot more information. We're getting the depth of the measurement, the quality of the measurement, the genotype, which is whether it's present in one or both of your chromosomes, the functional consequence, and the short name, which is an identifier from a public data set. And once we have that, we can do some queries in Zeppelin. Some SQL style queries to say, we're looking for genotypes or for variant observations where we have a good depth, so it's a solid observation, and that variant exists. So that variant exists in both the chromosome from your mom and the chromosome from your dad, and so these are the pretty serious variants, and so it just gives a researcher quick ability to look at that stuff in a very flexible way, using a language that they're familiar with. And of course, it's stored as an RDD. All of these result sets are stored as RDDs, or they can be converted to and from a data frame, which gives the users access to all of the rest of the algorithms in Spark, like clustering and that kind of thing. And so I think the last query that I want to show, this is a more complicated query. This is, again, satisfying that researcher's use case. I have a cohort here of a large number of patients, and I have a gene, FOXRED1, which is, again, associated with some really awful stuff if certain things go wrong in it. So let's do a list. Let's look in that cohort of patients. Let's find the homozygous variants and put that into a table. So that's exercising the seeking filter to go to Accumulo and skip through to those individual ranges for each of those samples. And we get a result back. And again, we can do some simple SQL queries on that data to see sort of where do those variants occur in the gene? Are they close to the start of the gene, close to the end of the gene? This is just kind of a fun example. I'm not sure it has any clinical significance, but you can see there seems to be, maybe there's a spike towards the end of the gene. Oh dear. I seem to have lost my... It doesn't matter. The last slide of my presentation was to say thanks. So that's pretty much the end of it. Those are the components of a pretty solid foundation for a genomics data warehouse built on Apache Accumulo. Accumulo was critical to the implementation of this. I can't imagine how we could have done it effectively in any other key value store, specifically the security model. The iterator stack, I think we could have done something similar with the iterator stack in another big table inspired key value store, but it was very, very clean to implement it in Accumulo. Again, the security model Accumulo was necessary for that. Yeah, it works really well, and I think it addresses both of those use cases really well, the doctor use case and the researcher use case. So I'm not sure how we are for time, but I'm happy to take any questions until Dan says I have to get out of here. None? Okay, well, thanks very much, guys.
All right, everyone, we're down for a 40-minute break. Please take your time.